Thank you very much, Alejandro. It's a real pleasure to be here, and thanks for inviting me. So I'm going to give you basically a tour of what we've been doing in Cork, right in the south of Ireland, and where we want to go with what we're doing. And I work in a medical school like many of you, and everything in medicine more or less goes back to Hippocrates. And Hippocrates is thought to have said, now it could be fake news, but he's thought to have said that all diseases begin in the gut. And that can be quite surprising if you start thinking about neurology and psychiatry. A lot of my neuroscience colleagues are often wondering why, if I'm interested in the neurobiology of emotions, why am I so fixated on what's going on in the gut? And all we have to do is look to our language. We use phrases like gut feelings, gut instincts. When we're disappointed, we're gutted. When we're brave, we make gutsy moves. When we are nervous, we have butterflies in our tummy. So perhaps there is an overarching or underlying neurobiology to these feelings. Now, every story, and I'm going to try and create this as a narrative, needs a hero. And this is the unlikely hero, perhaps, of this story. And most of you may not know who this is. All immunologists, of course, will know who this is. This is Eli Mechnikov. He was a Russian working at the Pasteur Institute in Paris. And he won the Nobel Prize in 1908 along with Paul Ehrlich. And he won the Nobel Prize for his seminal work in the discovery of phagocytosis, basically how immune cells gobble up foreign bodies. And, he was an amazing, one of the fathers of modern immunology. And as often happens, great scientists, later on in their careers, they can start coming up with crazy ideas. And Mechnikov was full of crazy ideas. And most of them are complete nonsense. But one of the things that he was fascinated about was, why did people in certain regions of Europe seem to live longer and live healthier? And so he focused a lot of attention in parts of what's now rural Bulgaria, and he wrote about all the different aspects of their life that enabled them to be actually have this increased longevity. And what he put it down to was what they ate. And what they ate was a lot of lactic acid bacteria from fermented foods. So not only is he the father of immunology, he's really the father of experimental gerontology in terms of really looking at how people age healthily, and also the father of probiotics, whatever that means. And so if you go to Korea, you can get a probiotic drink with his picture on it. So it must have that Nobel sign of approval, so it must be good for you. But most of what Mechnikov wrote about was ignored for at least 80 or 90 years. And we'll come back to him. So what we're really interested in understanding in our lab is really the relationship between the gut and the brain at all these key windows across the lifespan. And so we have ongoing programs in the perinatal period, which I'll touch on, in adolescence and in aging. And I just put this up here just to highlight that when we're talking about the impact of diet and the microbiome and the gut on brain health, it will be different depending on what stage in the lifespan you're actually looking at. And I understand in this project there's lots of different cohorts from different stages, so that's just worth remembering, because we know what's going on in the brain is different at these time windows. I need to introduce the microbiome, but I really don't, because most of you already know what the microbiome is. But there are a few things that I want to reinforce about the microbiome in terms of sheer numbers. We are more than if we look at our genes, we're more than 99% microbial. That's really humbling. If you think of all the money we spent on the Human Genome Project, it's for less than 1% of our genes. Sorry, Alejandro. That's really important. The weight of our gut microbes is roughly about the same as that of our brain, which is also quite humbling as a neuroscientist to think about. And the other thing is, when we talk about the microbiome, we, for the most part, and almost everything that you'll have heard of at this meeting, is not talking about the microbiome at all. It's actually talking about the bacteriome, because what we're measuring is just bacteria. But the microbiome is much, much more richer. It includes the viruses and bacteriophage. It includes the archaea, the fungi, all of which make this rich ecosystem, and most of which are completely ignored to date in relation to brain health. But there is a lot of really cool stuff brewing, and we're working on this as well. And this is a field that is changing all the time. Alejandro and I were just talking, what was best practice 2 years ago is not best practice anymore. So as young scientists navigating this, it can seem quite unwieldy to try and keep up with it. If I'd given this talk 2 or 3 years ago, I would have told you, and you see this in names of books, that we are 10 times more microbial in terms of cells than we are in terms of humans. But that has been revised downwards, and it's now thought to be about 1.3 to 1. So we're still more microbial, but just about. What we're really interested in trying to understand is what microbes are doing from the moment of colonization at birth all the way till the end of life. And really, for the most part, we're thought to be sterile and neutral, and we get our microbes as a kind of a birthday present from our mothers as we're on the day we're born. So as we come through the birth canal, we start the initial colonization, and these microbes then start to inform the developing immune system and really start helping programming many of the systems within the body. And so one of the big things in the field is to try and understand what are the factors that are influencing this composition in early life and throughout the lifespan as well. And here you can see many of these factors that have been really well studied. Diet, exercise, exposure to drugs, including antibiotics, the environment we live in, etc. I poo-pooed genetics, but actually there's a really understudying of the role of genetics and epigenetic factors in relation to host microbe interactions and trying to understand that. And one of the factors that we've been really interested in has been in relation to mode of delivery at birth. Because, of course, if you're born by C-section, you bypass this handover from, you get a different present, shall we say. So really, if we look at it from an evolutionary perspective, and I would encourage you to do that because microbes were here long before humans, and there never has been a time where the human brain has existed without microbial signals. And so during pregnancy, basically the microbiome will change and evolve and to be optimal to hand over to the infant at birth. Now, if an infant is born by C-section, this is different, and you kind of have it's like a relay race where you're handing over a baton from mother to infant overall, and this is bypassed, and the microbes of C-section born individuals are quite different to that than those born vaginally. This goes back to a very small pilot study now, which wouldn't get published today because of its small sample size, from Gloria Domingos-Bello's group now at Rutgers, where they show that vaginally born are separate very, very different from C-section born individuals. This has now been replicated in at least 14 other cohort studies. And so people are beginning to see that these microbes could be the infant's security blanket in early life. Perhaps the largest study is this TEDdy study of 12,000 individuals, which is showing very clearly that birth mode is a key factor in driving the changes in the microbiome in early life, as is mode of nutritional provision, whether an infant is breastfed or bottle-fed, and we'll come back to that as well. Even just this month, work from Trevor Lawley's group in Cambridge showed very clearly in Nature a big difference in the microbiome of infants born of C-section versus vaginally. And so that's something that also is reinforcing that we're switching this ecosystem in early life. Now, we know that infants born by C-section are through epidemiology, at a greater relative risk of developing allergy and asthma and type 1 diabetes. That's very well known from epidemiology. Now, the focus on the word relative risk. We're talking about small but significant sizes. And so we know very little about C-section and relationship to brain health overall. And so we worked with our epidemiology colleagues on this and got a lot of interesting, if somewhat conflicting data. But I'm an experimental neuroscientist, so we wanted to develop then an animal model that would allow us to actually challenge this and see what actually was going on. And this is work of Livia Murray, who's now a postdoc at Caltech, where we were looking at a mouse model of C-section, and we examined, well, what happens then in terms of the microbiome, and we can see that in C-section born individuals, there is a separation in early life. This is postnatal day 9. By adolescence, it starts to come together, but there's still a difference. But by adulthood, it's completely undistinguishable. So you can't tell in adulthood if someone was born by C-section or not. But what are the enduring effects of this kind of disturbance? And what we find is that even in early life, there are changes in anxiety-like behavior and an elevated stress response overall. When we look at things like social behavior, and I'll keep coming back to social behavior because it's one of the areas where we find when we do any form of manipulation of the microbiome, especially in early life, that we find changes in sociability. We're able to find that mice born by C-section are not able to distinguish maternal bedding from neutral bedding, or deficits in distinguishing a novel versus a familiar mouse. So it's telling us that there are these enduring effects, and we're looking at the mechanisms of this and trying to disentangle this. Of course, the important thing to remember is that C-section is a life-saving surgical intervention. And the last thing I want to be is a male on a podium talking about women's obstetric choices overall. So you need to realize that it's happening. In some parts of the world, it's happening at rates that are way, way, way beyond norms. In parts of Brazil, it's at 70%. In China, it's over 60% in parts. And even in Ireland, it's doubled in the last 30 years in terms of the amount of elective C-section. And people assume it's benign, but the question is, can we look at intervention strategies that will actually reverse some of these effects? And that's something that we're studying with some of our industry partners to be able to develop a microbiome-focused dietary intervention early in life that might actually foster re-equilibration of the microbiome. And that's something that we're quite excited about overall. So this was in mice. Mice are great. I love mice, but they're not going to, tell us much about. They'll tell us something, but they're not going to explain the whole story and complexity that we have in humans. So we wanted to look at this, in a human study, and we wrote a grant, and it was part of the My New Gut project, which was a previous EU consortium we were involved in. And we never expected this really to pan out. We wanted to see, well, what if we took, I was going to say normal, but Irish medical students, and stratified them by their mode of delivery. And so then we wanted to see, well, how could we ethically stress them in a way that would enable us to see how they dealt with stress. And so luckily there are some great ways of doing this. One is a Trier social stress test that many of you know. It's basically a public speaking test where people are brought in and are asked to pitch their dream job to these quite grumpy looking people in front of them, and there's a camera on them, and they mount a stress response, and we measure a variety of biological and psychological outputs in these individuals. And so what we found was if we stratified individuals based on their mode of delivery, we didn't find much in terms of cortisol, but in relation to the immune and psychological response to acute stress, we did find that those who were born by C-section had a greater psychological response to an acute stress. So that's acute stress. And acute stress is interesting from a physiology point of view, from a coping point of view, but it's acute stress. What about chronic stress? As most of you know, in relation to psychiatry, we're more interested in chronic stressful events. So could we ethically, chronically stress individuals? Well, the great thing about being an educator is that we do this all the time by making our students sit exams. And so we can monitor how they respond to exam stress period. And what again we found was that there were subtle but quite significant increases in the behavioral and immune parameters related to chronic stress. Most notably the Cohen's perceived stress scale, that we only saw a real increase in perceived stress in individuals that were born by C-section. So, and of course, this is not, C-section is more than just microbial changes. You lose the priming of the stress of being born, the immune priming, etc. But it gives us some idea that these studies that we have in mice can actually translate to the human situation. But for my skeptical neuroscience colleagues, that's not enough. That's kind of association, and it's interesting and it's nice. They need more evidence. So how do you get more evidence that the microbiome might be relevant to brain function? You can just turn to other areas of biology or engineering. If you want to prove that something is important, well, why don't you take it out and see what happens? And so the concept of being germ-free or growing up without microbes is not new at all. It goes back to Pasteur's time. Here's a sci-fi magazine from the 1920s with a germ-free gut man in it. Luckily, our germ-free mice look a lot better, although they have their own problems. They basically grow up in a bubble. They allow us to answer the question, is the microbiome involved, yes or no? But they lack clinical translation because there's really no human growing up in a bubble, except maybe the boy in the bubble that Paul Simon sang about in the 1980s, and most of the young people have no idea what I'm talking about, but that's fine. But it is really an important model system to allow us to answer this question. And we've used it a lot, going back over a decade now, as have many other groups, to basically implicate the microbiome in aspects of brain function that we would never have thought of. For example, we did an RNA-seq experiment, basically an unbiased approach, looking at what genes are changed in the prefrontal cortex of mice that grew up germ-free versus controls. And what we found was there was a huge increase in genes involved in myelination. This was specific to the prefrontal cortex and specific to males. And this was really interesting that we were able to look down the electron microscope and be able to show that there is hypermyelination in our germ-free animals. Now, hypermyelination doesn't occur that often in nature. We mainly think of myelination in the context of demyelinating disorders like multiple sclerosis. However, there are some rare autisms where hypermyelination occurs. Adult hippocampal neurogenesis. Again, we were able to show that the regulation of neurogenesis is completely out of kilter in these germ-free animals. And this is the birth of new cells in the dentate gyrus of the hippocampus of at least mice that may be relevant to complex cognitive processes, including pattern separation, as well as antidepressant function. A variety of neurotransmitters and changes in synaptic plasticity, most notably BDNF, in the hippocampus in these animals, changes in dendritic growth. Microglia, the brain's macrophages, in a way going back to Mechnikov, who discovered macrophages. Work from Marco Prince's group and Freiberg and others have also shown that basically without microbes, our microglia don't act appropriately. And then work from Sven Pedersen at the Karolinska has shown that the integrity of the blood-brain barrier is disrupted in germ-free animals. So basically, almost any process you look at in the brain, you'll find changes in some way in germ-free animals. A study just out last week in Nature from David Artis and Conor Liston has shown that spines, using 2-photon imaging, that spines formation in the prefrontal cortex are also altered in these animals. And here you can see, this is a very simple picture of a basolateral amygdala neuron. And the amygdala is very important for fear responses, for pain responses, for social responses. And here you can see there's a complete increase in arborization in these amygdala neurons. And this is very much like a chronic stress state overall. And what does this translate to at the behavioral level? Well, it translates to deficits in fear, and I won't go into all the details, it's all published data. So we have deficits in fear conditioning, deficits in visceral pain, increased visceral hypersensitivity, and when we look at social behavior, mice, like humans, are quite social, and so if you give them the chance to have time with a mouse or an empty chamber, they'll gravitate towards the mouse, but not if they're germ-free. And mice, like some humans, are a bit fickle. So if you give them the chance to spend time with a mouse they already know versus a new playmate, they'll gravitate towards a new playmate. But again, not if they're germ-free. We've been digging into some of the molecular mechanisms that might underpin this. This is a work where, again, we're using RNA-seq to look at the pathways that are dysregulated when we force mice into social interaction and when we look at what's going on in the amygdala. And what we find, for example, is that there is a deficit in germ-free mice here in red in the MAP kinase signaling pathway, whereas this is reciprocated by an increase in RNA splicing and an enrichment in RNA splicing, which is quite interesting because changes in RNA editing have been widely looked at in the context of autism. This is a recent paper from the Geschwin group looking at it in postmortem tissue. And so we're finding that there is a relationship between microbial signals, RNA editing events in key brain areas and social behavior. But not all of these dots have been joined up yet. But it is quite intriguing. So when we start thinking about it, if you stand back again from a big picture point of view, and you say that for normal social behavior, and if you're a mouse, you need to have microbes in your gut. Why would that be? And we know that if you look at some of the evolutionary theories in cortical development from Dunbar and others, that sociability promotes elaboration of the cortex. And so that's a lot of the anthropology, neuroscience intersections have been focused on this. And that's quite interesting. We've shown now that microbiome affects social behavior and the microbiome affects brain development. So can we close this triangle in some ways? And then you start thinking about, well, if you're in a crowded room like this, it's much easier for microbes to be spread from one to the other. And so you can start asking questions about, like, well, what's in it for the microbes? from an evolutionary perspective. And that's quite an interesting idea. And so Roman Stilling was a German postdoc in the lab at the time, and we did a lot of riffing on these ideas, trying to figure out, well, what does this really mean? What are the implications of what we're saying? And we collaborated then with Seth Bordenstein, who's an evolutionary microbiologist at Vanderbilt, and I put some of these ideas to Seth, and he told me that I was completely wrong. And every now and then, it's great to be told you're wrong, and that's why we need to collaborate with experts, and that's why in multidisciplinary fields we can't know everything. But the reason that I'm wrong is that I forgot something that I've already mentioned to you, which is that the microbes were there first. So that you can't separate, what's in it for the brain and what's in it for the microbes. The brain, there has never been a time where the brain has existed without microbial signals. We have co-evolved with our microbes. They're our friends that give us social benefits, among other benefits. And so once you start conceptualizing the microbiome in this way, you start thinking about things a lot different. And so if you look across the animal kingdom, and this is something we have in a review just out this week in Science, no matter whether it's in insects or hyenas or mice or baboons or primates, you start seeing, if you start tinkering with the microbiome, you can shift dynamics and social hierarchies and social living, but also the opposite. If you start changing social dynamics, you start changing the microbiome as well. And so it's an evolutionary wired process in terms of linking into parts of the social brain, most notably in the prefrontal cortex perhaps, that are quite adept at sensing signals and environmental shifts in the microbiome. Or putting it another way, that the microbiome is a conduit for how the environment affects the brain through an indirect pathway. And that's something that we're quite excited about. And where all this is moving to is really in positioning the microbiome in early life as a key important factor in driving the developmental origins of brain health. And so if you think about things in relation to the Barker hypothesis, where you position both the prenatal and postnatal environment as being very important for shaping later health. And then, of course, host genetics is really important in this. We posit that the microbiome would be a fourth pillar in this model or framework for understanding the factors that you can influence what's going on in later life. The other good news about it, and I'll come back to this again as well, is that unlike your genetics, which you can't do much about except blame your parents and your grandparents, the microbiome is one that is potentially modifiable. That offers some hope or some agency to the individual to shape that. So that's a lot of what we're doing in early life in trying to understand this. I'm going to move quickly to adolescence because I know that's what a lot of people here are also interested in relation to understanding adolescence as a key time window for when nutrition can play a role in brain health. And also, as any of you who have teenagers will be very aware, the anatomy of the teenage brain is very, very different. And my official title in the university is Professor of Anatomy. So I always like when I get a chance to put up an anatomy. And what we're really understanding more and more is that the teenage brain is really one that's under construction. And what we are actually thinking about in terms of our traditional views of teenage, I shouldn't even use the word teenage because the adolescent brain is evolving much later into the early 20s as well. And it is a particularly, as all of you know, a vulnerable time period for the development of many psychiatric illnesses, including impulsive control disorders, substance use disorders, mood disorders, and adolescent onset schizophrenia. So it definitely deserves a lot of attention in relation to brain health. And indeed, a lot of the focus in biological psychiatry in adolescence is focusing on the effects of a variety of insults, be it alcohol, stress, poor nutrition, drugs of abuse, and inadequate sleep on the brain. But all of these could equally have effects on the microbiome and how the microbiome signals to the brain. And this is really, really understudied. And we need to do a lot more in relation to this as well. And that's something that we need to more attention on. This is just some data we have some years ago where we depleted the microbiome from adolescence, and we found that there were sex-dependent effects on anxiety and cognitive behavior, particularly in males. Again, everything I show you, most of the effects are much more prominent in males, which is intriguing. This is a collaboration we published this year with Beatrice Pisani's group in Florence, where we looked at the enduring effects of stress in rats during adolescence. You find deficits in cognition, especially hippocampal-dependent learning. And when we fed these animals a diet that was enriched in omega-3 fatty acids and vitamin A, we were able to reverse quite remarkably the behavioral deficits that emerges due to the adolescent stress. But we also, and I won't even attempt to go through this, but we also found that it could reverse many of the effects in relation to what happens on the microbiome. Now, this is correlative. We can't say that one is relevant to the other, but it is quite interesting overall to show that there is this clear relationship between diet, microbiome, and adolescent brain health. In relation to alcohol and other drugs of abuse, the field is starting to take off. This is some early work we did with Andrew Holmes' group at the NIH, where we looked at the impact of vaporized alcohol on the microbiome, and we find that there is a shifting in the microbiome towards a more pro-inflammatory phenotype, and work we did with Ben Botrell's group in Lausanne in an animal model of alcohol seeking where there's no actual alcohol on board at this point. We find that there's a relationship between the composition of the microbiome and dopamine D2 receptor expression in the striatum. So again, we're beginning to really just look at, from substance use disorder point of view, relationships between the microbiome and behaviors that are relevant to reward seeking and other aspects of addiction, which may also be relevant for food addiction, obesity, and other things that might be relevant to this compulsivity as well. So I've mentioned, well, actually, I didn't really go into it too much in today's talk, but we've shown a lot of data that stress, especially in early life, changes the microbiome in general, and that basically mice lacking, the germ-free mice, they have an exaggerated stress response, changes in their neurodevelopmental trajectories, alterations in behaviors relevant to cognition, anxiety, and social behavior. So the question next is, can we modulate the microbiome to have beneficial effects on brain health? And that's kind of the big question for the whole field. And of course, we start in animals, and this slide summarizes some of the really cool data that we have found over the years. What we found is that if we focus, for example, on specific strains of bacteria, Lactobacillus rhamnosus and biflongum, for example, that they reduce anxiety, have an antidepressant-like behavioral effect, enhance cognition, reduce the stress response, and change both GABA and serotonin neurotransmission in the brains of mice. We also have really nice data on prebiotics, in particular galacto and fructo oligosaccharides, GOS and FOS, showing really similar effects. I'll talk a little bit later about the short-chain fatty acids. And then more unpublished work we have, which I'm also really excited about, because again, it goes back to what Metchnikoff was talking about, is that if you look at kefir, and milk kefir, which is a fermented food, and we can also modulate behavior and brain changes in mice, which was quite remarkable. So, but what this slide doesn't tell you is that for the most part, almost everything else that you look at, we've looked at in relation to the microbiome does diddly squat, does nothing. So most strains of bacteria will do nothing. And the field is really good at lumping the word probiotics, using it as a catch-all phrase that all bacterial strains that might sound similar names are all lumped together. And we would never do this in pharmacology. We would never lump every opiate together and say it's going to have the same effect, because some of these opioids, like loperamide, won't get to the brain. But in this field, everyone just lumps things together. And so the questions we have for the field is really what to dig down at a granular level of what is it about these two strains of bacteria? What are they producing in terms of metabolites? What is decorating their cell walls? How are they interacting with the immune system? How are they interacting with other host cells? How are they sending signals to the brain? What is they doing versus ones that do diddly squat? So that's really important for the field to really look at, because and I'll come back to that if we have time at the end, there's a lot of misinformation out there. We need to really understand this. So I don't like using the term probiotics so much, because it has connotations for things that have no evidence that they're going to do anything. Because in Europe, by definition, a probiotic is a live bacteria taken in adequate amounts that confers a health benefit. Now, people forget the last part of that. So it's to confer a health benefit. To prove that something confers a health benefit, you need to have clinical trials that show this. And for the most part, this isn't the case. And in North America, it's the wild, wild west. And you can get away with anything and say anything. And so it doesn't help the field. And that's something that perhaps that one of the things you can look at in your consortia is how do you get information across to a public that is blinded by a lot of misinformation in this field. So that's where we are with that. Then I'm going to move to aging, the other extreme of life from early life. And aging is having marked effects on the microbiome, and this has been implicated in many aspects of aging, specifically frailty, sarcopenia, the cardiovascular dysfunction, but also in terms of brain overall. And this goes back again to Metchnikoff. I put up here a study from my colleagues in Cork some years ago now, where they took about 180 elderly people. This is from Paul O'Toole and colleagues, and they followed them over time, and they showed that their health outcomes, in particular related to frailty, correlated with the diversity in their microbiome. So those who had the most diverse microbiome had the better health outcome. So, well, what's driving the diversity in the microbiome? And that is driven by diversity in the diet. So this really helps to the field to realize that increasing diversity in diet will increase health outcomes through the microbiome, at least in some ways. And so they found that people who lived in nursing homes, which had very repetitive, white, bland, beige diets all the time, that they had the worst health outcomes and the least diverse microbiomes, whereas elderly people living in the community, eating a lot of vegetables and fibers that had rich diversity in the microbiome, had better health outcomes overall. And so there's lots of excitement in the field, but of course, as I say, it's nothing new. Metchnikoff was talking about this 100 years ago. But in relation to brain health, there's less on this, and so we started revisiting this, taking aged animals and looking at what's going on in relation to their behavior, and we find anxiety, deficit in sociability, and memory deficits, and we're able to look at changes in what might be driving this, and we find that there's increased intestinal permeability that's exacerbated by stress. We find there's increased inflammation, as well as changes at the microbiome level. And so the next question is, could we do anything about this? Could we target the microbiome in aging and see what would happen? And we have two strategies for this. The first one, I decided to focus on midlife for reasons that are obvious, because it's something that I'm very aware of right now. The second reason is that it's probably a time window when we have more probability of slowing something down and promoting healthy aging. And so what we found was, in middle-aged animals, if we look just at the brain, we find that there is increased neuroinflammation going on, driven by increased stress-induced immune priming, infiltration of monocytes, inflammatory monocytes into the brain, as well as activation of microglia. And so we can see this really, inflammatory effect. And then we decided to feed these animals a prebiotic, which is an inulin diet. And inulin is a very well-known prebiotic in lots of vegetables like artichokes and leeks, but in many vegetables. And we could find that we were able to dampen down this neuroinflammatory response, these persistent effects of aging through dietary means and by targeting the microbiome. And it also shifted the microbiome. Didn't reverse it, just shifted the microbiome in a different way. So what this means that if you're ever thinking of having a midlife crisis, that instead of maybe getting a motorbike, that maybe you should think about getting some more leeks into your diet overall. And this is Marcus Boehm, who led this study along with my colleagues Ted Dinan and Harriet Shillekins. We have ongoing studies right now where we're looking at transferring the microbiome directly from young animals into older animals. And some of the data is actually quite promising as well. So there are different approaches to look at this. But the big issue we have in the field is really, how is this happening? How can microbes in the lumen of the gut send signals to the brain to modulate complex aspects of behavior? And we're really only at the beginning of figuring this out, even in animals. And so it requires a lot more attention. What we know is the immune system is very important. We know that the enteric nervous system, the second brain, is also critical. And a variety of microbial-based metabolites can interact with different pathways from the lumen of the gut to the brain. Among these is the vagus nerve. The vagus nerve, vagus, it comes from the Latin for wandering. You heard of vagrant. Well, vagus is one of the cranial nerves, and it sends bidirectional signals from all of our organs to our brain. It basically helps us to feel how we feel. And so some years ago, together with John Bienenstock's group in Canada, we looked at what would happen if we cut off the vagus signals in a mouse, and would it affect any of the behavioral consequences of giving a lactobacillus? And to make a long story short, all of these effects were gone in vagotomized mice. So this tells us, as I like to remind people, that what happens in vagus doesn't just stay in vagus, but will actually affect our emotions in certain ways. And at least this is one of the conduits we have. What we're trying to do now is to dissect out these pathways in a circuit-based neuroscience approach. So one of my postdocs is really trying to pin down where in the nucleus tractus solitarius, in the brainstem, these signals are emanating on, and then how do they impinge on limbic circuits that are underlying the behaviors that we're interested on, using opto and chemogenetic approaches. But it's really exciting area, and it's something that we're quite interested in. The other important thing to remember about the microbiome is that the microbiome is like a factory producing all kinds of weird and wonderful chemicals that our bodies wouldn't have without it. We ignore this a lot of our times. So, for example, short-chain fatty acids, butyrate, acetate, propionate, we cannot make these. These are only made from microbes which ferment fibers. So in a kind of a different approach, what would happen if we looked at the impact of these short-chain fatty acids themselves on behavior in animals? And we were able to really dampen down a lot of the enduring effects of psychosocial stress by using this approach. And this is known as a post-biotic approach. So you're basically avoiding the microbes, but just going to the actual metabolites that they're doing. And this offers opportunities then for companies to look for new ways of generating different metabolites that might have long-lasting effects overall on brain health, etc. And we're beginning to understand that many of these metabolites from microbes act on G-protein coupled receptors in the lumen of the gut, on epithelial cells, on nerve cells, in the enteric nervous system, on immune cells, and potentially even if they get to the brain in adequate concentrations, which is still one of the big problems we have in trying to decide, also in the brain overall. And perhaps the best example of this is in early life nutrition. The best example of understanding how microbes are so important in terms of shaping the impact of nutrition on health. Because one of the amazing things that, you know, in the microbiome field, it never stops amazing you. You find all these amazing stories about microbes changing host behavior in different ways and whatever else. But one of the most amazing things for me on this journey has been when I realized or was told that human breast milk contains the highest complexity of sugars of any mammal, about 20 times more complex than bovine, than cow's milk, which we use in our infant formulas. The thing about these sugars is they cannot be broken down by the infant. We have no capacity to break down these sugars. Now, why does evolution give us something so rich and so complex without giving us something to actually work with? And in fact, it does, because these are the substrates on which microbes act to produce key chemicals. And the microbes break them down. And so the bifidobacteria in early life act on these human milk oligosaccharides to produce chemicals like sialic acid. And sialic acid is really important for brain development. And so we're beginning to understand that, you know, we have co-evolved with these microbes to help us benefit as much and from an energy perspective, get the most out of the nutrition that we have. This is an example in early life, but why would this be specific to early life? This should carry over the whole way through the lifespan, in that in order to benefit most from the diets that we take and that you'll construct and test, we may need the appropriate microbes there to get the best from it. And if we don't have the microbes, then we're basically flushing things through this inner tube without getting any of the benefits from it. And there's a large inter-individual variation on that, and a lot of excitement in the field in terms of trying to understand this very complex relationship between diet and gut microbes overall. And what I'm just saying is that can have long-term effects in relation to brain health overall. All of what I've told you then leads to this area that we have coined psychobiotic. We've modified this definition over the years. Basically, it's targeting the microbiome for mental health benefits. And so we feel there's a huge need now to move away, to build on the animal work that we've done and move towards human translation. And that's where we're going. And these are some of the approaches that could be used in as psychobiotic therapeutics. I've talked already about probiotics and fermented foods, and prebiotics, which are basically the fertilizer on which these beneficial microbes thrive on. Then we have synbiotics, which is a combination of prebiotics and probiotics, which are widely used in the infant formula field. Paraprobiotics, that might be a new one for you. That's basically a dead bacteria. And that might seem counterintuitive, but one of the things we need to know is that bacteria can have basically sugars or various pili on their cell walls that can interact with the host immune system in different ways and stimulate that. And so in a way create a vaccine type of approach overall. And I've mentioned postbiotics. Okay, so this is Tracy Chapman, and it's 30 years since she was singing about fast cars and talking about a revolution. And in that 30 years, we've had very little innovation in relation to how we manage mental health. Perhaps it's time for a new revolution, and we've coined it the psychobiotic revolution. And so perhaps it is time that we move, thinking about the role that nutrition could play in shaping our microbiomes, in shaping brain health. And so we wrote this book as a way for the lay public to really think about this in a different way. And revolution is a grandiose term. We're only at the beginning of it, but revolutions have to start somewhere. And that's what's so great about this consortium and other such efforts, because they are really positioning nutrition at the heart of research in mental health. And I think we still have a long way to go. So do we have any evidence? Well, this is some evidence from our own work. It's a small study from a few years ago. This is Andrew Allen, a psychologist on our team. And what we did was we took one of the bacteria that we came through our preclinical pipeline, a biflongum 1714. It had great effects in mice. And so we want to see, what would happen if we took it in our human platform. And so we took healthy, normal people, well, Irish students, and we brought them into the lab and we stressed them as we do. And we found that if they'd taken this product, they had a reduction in their stress response, and they had a EEG signature we could detect, and there was a CANTAB on the Cambridge touchscreen platform. We could find that there was a signature in relation to working memory on the paired associative learning task. More recently, work from Paul Inck's group in Tübingen has used the same strain of bacteria, and they put people in the scanner, put them through a cyberball task. I don't know if the cyberball task is basically where you play a ball game with two other people in virtual reality. You're basically passing the ball around to each other, and all of a sudden you in the scanner gets excluded, and the other two people start playing, and you get ignored. And your brain doesn't like being ignored, and it gets frustrated, and there's a very well characterized signal of how this occurs in the stress-like situation. And in the individuals that had taken the biflongum, they didn't mount the same response. So again, we're getting validation. So that was really exciting for me as a basic scientist to be able to get any signature at all, that any of this could translate to humans, was fantastic. And we were really excited. So then we decided, let's take our L-rhamnosus. It's the best one we have in our animals. Let's take it. We've got great data. We know it's through the vagus nerve. We know what's going on. Let's put it through the exact same paradigm. You cannot get more negative data into the one paper if you tried. It did nothing. It did absolutely nothing. And boy, did we try, because we had an MD on this project and he really wanted positive data. So, what this highlights is that there are limitations to all we do. And this is not specific to the probiotic field or the microbiome field. This is in all aspects of translational neuroscience in general. And it's something that is very important to highlight overall. Where the field is going, I really like the study that we wrote an editorial about from Joran Reis' group in Leuven from earlier this year, where they looked at over 1,000 people and looked within them, looked at their microbiome, and were able to show within them those that had depression had a different microbial signature, or a different enterotype, as they like to work with. And then they mined the neuroactive potential of these microbes. They used shotgun sequencing to be able to look at what actual potential metabolites could they produce, and created a gut-brain module literature, which was really, really nice, and identified where the actual microbes could impact brain function. And what's nice, then they had a validation cohort from the Groningen Lifelines Deep cohort with Sasha Garnikova, and so they were able to validate. In that cohort, they didn't have depression, they just had quality of life scores. So it's not really, but in the Leuven group, they did have GP depression validation, and then they went into a small group of treatment-resistant depressive patients and were able to validate it there. So this is where the field is moving, into large-scale studies where you're able to drill down into mechanistically what's going on, but it still is only correlational. We need much more on the longitudinal, and to see how symptoms may relate in time to some of these effects that are ongoing. Okay, no talk in the microbiome field is complete without mentioning fecal microbiota transplantation. Taking someone else's poo. And so this is really disrupting our whole concept of what medicine is. Overall, it's challenging regulators, and it's emerging from the infectious disease field, where a number of years ago in the Netherlands, they showed that individuals with C. diff, when they got a Clostridium difficile infection, when they got a fecal transplant, there was a 90% efficacy. Now, in medicine, we have very few examples of where there's a 90% efficacy. And so in doing so, then people are now trying to see, will this have benefit in other syndromes like inflammatory bowel disease, irritable bowel syndrome, metabolic syndrome. There is one pilot study in autism as well. So it's really a growing field. We've been interested in this, and so what we did was we looked at the microbiome of depressed individuals versus healthy. We find there's a decrease in diversity, and then we took that microbiome and transplanted it into rats, in this case, and we were able to emulate many of the symptoms of depression, including increased anxiety, anhedonia, and inflammation. And this is something that people are using now to help build causality, going back to what we were talking about earlier in relation to what Clara was saying, to try and validate what's going on. It's been done now in autism, in ALS, in Alejandro's data is in ADHD. We're seeing this happening more and more, that you're able to have very distinct effects being transferred from humans to animals. And it's outlandish, but it helps us show that there's something in the microbiome that is signaling to circuits in the brain that are mediating these behaviors overall. And so we're beginning to see for depression that if we contextualize depression as a disorder of stress, but it's a disorder of stress along with a disorder of the inflammatory system coupled with changes in the microbiome, that it creates a holy trinity. So this is a way of looking at it, and that maybe to treat it, we need some form of modality that will tackle all three parts of this, and that's something that we're quite excited about, but we have a long way to go. And while this seems really new and outlandish, and psychiatry is, has a kind of a back off kind of view on it, it's not new at all. If we had been reading the British Journal of Psychiatry in 1910, we would already have heard about the treatment of melancholia by lactic acid bacilli. So while those of you early career people who think anything before the year 2000 was old, there's a whole literature even before PubMed. And I would encourage you to think that we're not so smart in today's society. We have better technologies and we have better resources, but in terms of the concepts we have, many of them are out there from an older literature, and we have to just apply them in a way that will be very relevant overall. The last thing I wanted to mention, but just because I know some people will be interested, is the whole area about how the medications that our patients take, the medications that we have, how they are interacting with the microbiome in a way that we really never understood before. And that perhaps the microbiome is this kind of chamber of secrets that unlocks both the efficacy and potentially the side effects. The whole area of pharmacomicrobiomics is huge right now in oncology and in metabolism. So we now know that checkpoint inhibitors, which are these drugs to treat cancer, melanoma in particular, that the efficacy of these checkpoint inhibitors is dependent on an individual's microbiome. And if people are taking antibiotics, they're not working. And so that's really, metformin, how metformin works in diabetes is also been shown to be due to what's going on in relation to the microbiome. There's ongoing work in everything from paracetamol to digoxin. So why should psychotropics be different? And they're not. And we published many years ago now in showing that antipsychotics, they change the microbiome quite dramatically. This is work on olanzapine in particular, but we also have unpublished data on risperidone, and that if we tinker with the microbiome through antibiotic usage, we could actually shut off the weight gain associated with antipsychotics. So 1% of the population are on antipsychotics, and most of them are developing weight gain. That doesn't make sense from a pharmacology perspective based on the receptor profile. And we think the microbiome is really important for this. Sofia Cosoto just finished her PhD in our lab, and she expanded these studies looking at 7 other drugs, from antidepressants to mood stabilizers. And she was very excited about this because she did it both in vitro on 2 different strains of bacteria, in vivo, and looked at gut function. And we were able to show that in particularly fluoxetine, lithium, valproate, and aripiprazole were having marked effects on the microbiome. And so she thought she was doing great. 6 drugs, different modalities. This will be a really nice paper. And then this paper came out. And this paper from the Embel group in Heidelberg had 1,000 drugs across 48 strains of bacteria, but they didn't do any in vivo data. But the nice thing is, even in the abstract, they say that particular classes, such as chemically diverse antipsychotics, were overrepresented in the group. And basically 1/4 of all drugs in pharmacies are interfering with the microbiome. That's remarkable. And then perhaps if haloperidol was discovered today, it wouldn't be discovered as an antipsychotic, but as an antimicrobial agent, because it has marked antimicrobial effects. And what we label drugs may be quite different. So there's a lot of really excitement going on in this field. The other area that's really exciting right now, relevant to this consortium, is in trying to understand the impact of modern lifestyle and diet on the microbiome. So people are going out and looking at ancestral microbiomes in people in Tanzania, for example, where they have still have hunter-gatherer populations in the Hadza community. They have a very rich, diverse microbiome, largely driven by their fiber intake. We can then see what agricultural practices has done to the microbiome by looking at modern-day Venezuela and Malawi, and you start seeing there's a diminution in the diversity of the microbiome. And then finally in our Western society, and it's the US, but it's all over Europe and everywhere else, with the introduction of processed food, with the introduction of antibiotics in the food chain and the overuse of antibiotics in medicine and the stressful life that we lead, we have lost the microbiomes that our ancestors have. So one of the questions that remains, and it's quite a lot of really exciting work from Justin Sonnenberg and others, is what if we then started taking these diets? So shouldn't we just take these diets? But the problem is if you don't have the microbes to harness the benefits from the diets, it's completely waste of time. And so there's a real lack yet of understanding of how we could actually change this diversity problem overall. And of course, we don't know how to harness this in relation to mental health at all. Because in these populations, there is no multiple sclerosis. There is no inflammatory bowel disease. There's no inflammation. These are disorders of the microbiome, disorders of Western civilization, largely thought now to be the microbiome playing a key role. But we know less about it in the context of brain health or even in disorders like autism. We're working on this. This is work that Kirsten Birding-Harland, postdoc in the lab, is really trying to do. We're trying to develop a educationally informed psychobiotic diet that is really rich in fermented foods and fibers that are going to promote the beneficial microbiome and help us really triangulate the relationship between diet and brain health through the microbiome and trying to understand that. It's just at the beginning. We have very, very only pilot data right now, but it is an exciting area where we're moving into the future to actually see what we're trying to do is over 1 month in people with relatively bad diets, can we shift their microbiome and see how they deal with stress in the laboratory environment? And hopefully that will work. And that will allow us to test whether our state of gut will markedly affect our state of mind. I won't go on too much, but this is just some general things about the microbiome that are worth mentioning. The big problem is there's no such thing as a normal microbiome. You can go to your doctor and get your blood pressure checked and get your cholesterol checked, and they can tell you exactly where you are under physiological norms. You go in with a sequencing profile of your microbiome to anyone, and they'll say, that's interesting, but I don't know what to do with that. And that's not just educational, it's just that we don't know what it really, we know big picture changes at a level we don't know, but it's changing. Sequencing costs are coming down, longitudinal measures are happening, people are really looking at personalized diets and interventions. We're beginning to understand the relationship with host genetics and the environment in general. I put this in for buyer beware. There's lots of really bad stuff out there. If anyone's trying to sell you anything on the microbiome, there's probably something, we probably don't have enough evidence right now. So be careful. Always ask, well, where is the clinical evidence of what you're actually trying to sell on this? If people are making money, it's not clear where that is, where the data is. And it's not just in terms of corporate selling. There's also a communication misalignment. This, for example, is a report in the BBC last year from a really good journalist, James Gallagher. He did really brilliant work, and I'm not sure he's responsible for the headline. It's a study from Israel where they looked at, they took probiotics from a health food store that have never been shown to do anything, and they gave them to humans, and they didn't do anything. And so then there was a big story that probiotics labeled quite useless. But again, going back to my lumping idea, no one's ever showed that these specific strains did anything in the first place. So we need, some level of countering this through the media, but we have a long way to go. And as I mentioned, unlike our genome, our microbiome is potentially modifiable. These are some of the things we try and, that there are some emerging data, or at least good data, to say factors that will increase the diversity of your microbiome and promote beneficial microbes from a dietary perspective. It's prebiotics, omega-3s, polyphenols in particular act as really great agents to modify the microbiome, and the microbiome modifies the activity of polyphenols and flavonoids. So there's a lot of excitement in that field. Again, decreasing intake of processed food, emulsifiers, sweeteners, exercise, microbiome interactions are really important. And that's lots of excitement in that field. Sleep, circadian rhythms and microbiome is also brewing right now. Avoiding C-section as much as possible and supporting breastfeeding and minimizing antibiotics and eventually weak cage in our house. But having a pet is perhaps one of the best ways to increase your diversity, particularly a dog, not a cat. There is good data now in epidemiology studies from the States showing that. The other concern we have in this field is hype. And for young people, this is really important just to point out, you have a normal hype cycle about different things, whether it's, right now in biology, it's single cell everything. Jonathan Eisen at the UC Davis gives the Overselling the Microbiome Award. This is not an award you want on your CV. The challenges are really about going from rodent studies where there's these remarkable effects where we control everything all the way to humans. But the big one is correlation does not mean causation. And the microbiome field is the most guilty of this overall. There's a great website called Spurious Correlations. For example, you can correlate the divorce rate in the state of Maine with the per capita consumption of margarine. That gives you an example of some of the caution you need in this. Now, I'm Irish and we like butter, and you can come up with a story if someone came home with margarine instead of butter, that you could start a row and that could lead to a whole load of things. And that's where this field is going, because it's looking at one change in one bacterial strain in one situation, and it's not putting everything in an ecological framework that makes sense. Lots of small underpowered studies. Lack of dietary information is key. It's a big problem in the field. Everyone thinks they can be a psychiatrist if they can do a few scales, but no one wants to train to be a psychiatrist. So good psychiatric phenotyping. I mean, this is really relevant in this field where a lot of the studies are done by gastroenterologists and immunologists who don't know anything about how to phenotype from a psychiatry point of view. And I think this is really where psychiatry as a discipline can really come into its own overall. And we need rigorous human trials with all of these interventions across the lifespan and really dig into mechanism overall. So this is the work of a large team of us in Cork. My really good clinical colleague, Ted Dinan, who's just retired as the head of psychiatry. And we're back to Hippocrates. Remember Hippocrates? Well, one of the other things he's supposed to have said, again, it could be fake news, is that let food be thy medicine. And so we might change that to let food for your microbes be your medicine. And I leave you with this cartoon that the dreaded Daily Mail made about our work. So thank you very much. And sorry for going on a bit long.